Hello, my name is Michael Jones. I'm a principal scientist here at Waters Corporation, working in the Pharmaceutical Life Sciences Laboratory. What I'd like to speak to you today about is investigating the benefits of implementing what convergence chromatography can mean for the medicinal chemistry and process development laboratories. So with synthetic laboratories, they can look at a wide range of different types of products. They may be making different chemical materials like APIs and so forth, or they may be doing different types of monitoring of reactions. They may be looking at a whole synthetic route within a particular reaction. About 6 months ago, they sent me out to a collaborative laboratory, and we picked out 3 different drugs that we wanted to synthesize. We wanted to test out our technology. We wanted to make sure that we can actually get some benefits by putting this new technology within a particular medicinal chemistry or synthetic type of laboratory. The types of benefits that we wanted to explore were what does it mean if we couple the convergence chromatography system, the UPC squared system, with mass spectrometry? Is selectivity going to play a role with this particular type of technique, with this particular type of application space? We wanted to explore also chiral compounds. So the compounds we chose had a certain chiral property. We wanted to look at the enantiomers, whether it be enantiomer excess, or just looking at whether or not we isolated the right R or S form of that particular drug. We also wanted to compare this technique with UPLC, or reverse phase liquid chromatography. We wanted to see, are we getting any extra benefits from that approach also? So within this poster, we tried to highlight some of these benefits. And you can see, one of the first things we noticed right away on one of these reactions, it was a reaction step, we called it reaction step 7, for a drug called Crestor, or rosuvastatin. And you can see, in this instance here, we can separate the product and the starting material of that particular reaction, but we can also see, aided by the mass spectrometer, the presence of some impurity byproducts of one of the starting materials. Now this is important to realize because if we don't control these byproducts, they may affect the reaction and the progress in terms of yield and purity later on when we get to the final product. Looking at the different type of selectivity, I mean, we did launch out different columns with this technique, and we wanted to see, well, what does that bring to this application space of these synthetic chemistry workflows? And we can see with the UV data and also the MS data, we see a lot of different things going on as we screen through the columns for this particular reaction. We can see in some instances over here that we may get added resolution between a couple peaks. We can also see in some instances where you might get selectivity changes. In some instances, we also saw, again, a peak in the mass spec, but not in the UV because it was lacking a chromophore. So with all this information together, we can make better decisions as to how best to progress this reaction or progress this synthetic route later on to its next stages. This other example, down below, we looked at the at these chiral components. It's a reaction for clopidogrel. It's also marketed as Plavix. In this reaction, you can see when we analyze on one chiral column, we can see the separation of the starting material enantiomers, but not the product enantiomers. But yet, when we go screening to the next column, we can see the separation of the product enantiomers, but not the starting material enantiomers. In many instances, the people want to know everything about their process. So choosing one chiral column, which maybe they found during the stage that brought them the chiral starting material, and as it went into the next stage, that chiral column wasn't going to work to separate the enantiomers for that product of the next reaction. So it's always good to have a system that's set up in a way it's flexible enough to have different chiral columns. We also wanted to explore the use of this technique versus what's traditionally used today, which is a reverse phase type technique. They might be using HPLC, some have progressed to UPLC. In this instance, we can see, as we compare over here, on this, we revisit the Crestor reaction, we look at time point 0, and we can see this nice separation of the starting material and one of the other starting materials, so before the reaction has begun. When you compare the UPC squared results with what some people do, which would be a low pH screening and a high pH screening on their reverse phase system, you can see, although they get a selectivity change, they don't get a great separation between those two components. Now, before the reaction's begun, we don't know if that's going to cause itself a problem or not. As we look 6 hours into the reaction, we can see that as the product grows within this reaction, we get a very nice separation with a lot of resolution between the starting material 1, starting material 2, and the product on the UPC squared instrumentation. But as you can also see on the chromatograms below, the low pH and the high pH UPLC data show that you get a coelution between the product and the starting material, which is starting material 1, and we don't even see any response for the starting material 2. So there's a sensitivity challenge for that particular technique. By looking at these results, we realized that having a UPC squared type technology and a reverse phase technology are complementary to each other, and that we need both of these technologies within the laboratory. So there are some other key instrument capabilities which we really can't show with chromatography. And those capabilities we tried to pull out in terms of points of note. We can see that this technology, UPC squared, it was like stationary phase agnostic. I could stick any column on there. I can stick our UPC squared branded columns on there. I can put chiral columns on there. I could even take any type of reverse phase, Hillock, or normal phase type of column and use those as selectivity tools to help me manipulate my chromatogram to find those impurities which might affect the actual reaction in the progress of its lifetime. When I was doing the chiral type screening and achiral screening, and you might have seen this in another video by a colleague, Sean McCarthy, we had independent temperature control of each column bay. Why that's so important is because a lot of the initial steps, they will show that the achiral columns, I might use a higher temperature, 50, 55 degrees. Where the chiral columns, they have upper temperature limits of like about 40 to 45 degrees, typically. And because I can control those temperatures independently within this particular instrument, I don't have to worry about damaging my chiral columns if they're in the same bay of a typical column heater that you might find anywhere else in the market. So I've now got a technique that prolongs the actual consumables that I have within the laboratory. Another point is, for reaction monitoring or process development, when you're doing some of this reaction optimization, the samples that you're going to be getting are primarily organic. And you can take aliquots and you can dilute them up with a little bit more organic if you wish. You can directly and shoot them if you want at a lower injection volume. But being able to inject samples of 100% organic was very beneficial because I did not have to convert that to a more reverse phase friendly type of technique. That's typically where they might do a sample preparation that includes a more aqueous environment, assuming that solubility for them allows them to do so. So if you're thinking about implementing this technology within your laboratory, perhaps you'll see the benefits that we've found in terms of how it was coupled with mass spectrometry and seeing peaks that you don't normally see with just UV, to altering your selectivity with this stationary phase agnostic approach, whether you're using Hillock, reverse phase, normal phase, or our UPC squared branded columns. The chiral aspects, in terms of maintaining the lifetime of these very expensive chiral columns with this independently temperature controlled system. Or perhaps it's just having an orthogonal approach within your laboratory, something different from reverse phase chromatography that will help you prove the purity of your compound as you move it forward to your compound library. And with that, I thank you for your attention.